on the transcription administration and transcription factors. This lecture will be given to you by Dr. Aurelio Balsalobre, who is a research associate here at the Institute in the department of the unit of molecular genetics. So he is a specialist in genomics and transcriptional regulation. So I think you should learn a lot about all these, and he's the perfect person to give you this lecture. So enjoy your lecture, and thank you.
So the lecture will be given in English, but you can, first of all, you can stop at any time. Okay. We have 2 hours, plenty of time, so I can take an aparté to give more details or discuss something that you didn't understand. You can ask your question in French. I mean, French is my first language, so you can ask the question in French, and I will answer in French as well. So the purpose of the lecture today is you are all coming from a different background, and it's time to have a kind of summary and facts about transcription, how it works, what are the actors involved, how transcription factors is regulated, and what's its importance. And so the first part of the lecture are like data, I mean theory, and then the second part I will use the story of the lab I'm working in. So it's the lab of Dr. Drouin here at the Institute, who has been working on the same promoter for the last 25 years, and I will use the history through the key experiments that have been done to see the experiment that can be done today, how also the field has changed throughout the decade. So first, where does transcription stand in the life of a cell? So transcription, it helps to make, I mean, it's in the flow of information from the DNA to make a protein. Oui? Moi, c'est ce qu'on m'a demandé, c'est de le donner en anglais. Mais tu peux, n'hésite pas à m'arrêter quand tu veux, et même après le cours, si tu as besoin tu veux un petit peu plus de temps, là, il n'y a pas de souci, je peux rester là aussi. And so, actually, in the eukaryotic cells, the DNA is stored in the nucleus, okay, which is about 1 micron. And the whole DNA has to be packaged to be protected. And the proteins are produced in the cytoplasm. So you need an intermediate, the mRNA, going through the nucleus to the cytoplasm. This achieves also an amplification step, where with 2 single strands of DNA, you can reach millions of protein per cell. And it is the regulation of the transcription that will allow differentiation. So with the same material, genetic material, every single cell of your body has unique properties, not only the shape, but also the metabolic enzymes, the all the receptor to respond to different stimuli. Okay, so all of this is specific for each single cell, and that allow your body to work as a team. Okay. Also, I put a new slide this year just to make sure that we are all talking about the same language. Okay, when we are talking about transcription, so the enhancer is DNA sequence that can be upstream, downstream, within the gene, but this sequence can be moved away. Okay, it can be, and this sequence helps the regulation of the promoter. Sometimes you can have an enhancer that is really very far away, like a megabase, with several genes in between, but the interaction enhancer promoter will be specific. So one enhancer will specifically activate or repress the transcription directed by this promoter, even if you have 2, 3, 4 different genes in between. Then you have different exons, different splicing depending on the exon that are used. Then you have an mRNA. The 5' UTR are the sequences that are before the ATG. Okay, this stands for untranslated region. Then you have, let's say here, an ATG. So this will be the coding sequence. Then you have a TGA to end the protein. And so you have also a 3' UTR. Usually, the 5' UTR are quite small. Why? Because usually you have scanning of the mRNA and the protein start with the first ATG. So that's why the 5' UTR usually it's a short sequence. Maybe 100 base pair, sometimes even less than that. Although the 3' UTR sometimes can be very long, KB sometimes. Okay, you may have, let's say, 5 or 300 base pair of coding sequence, and then 1 or 2 KB of 3' UTR. And this is a style for multi-regulation, transcriptional, post-transcriptional regulation. And then so you have a protein. So transcription. So you start from DNA, okay, and you are going to produce an mRNA. And in this lecture, we'll just talk about mRNA. It's not the only one. You have ribosomal RNA, tRNA, microRNA, but we will talk only about mRNA in this lecture. So all of these are produced by the RNA polymerase II. There are 3 RNA polymerase in eukaryote, and here you need the 2. So first of all, you have to find a spot on the genome to start your mRNA. So it's called the initiation, okay, where the mRNA starts. But before that, you have a step which is called as a pre-initiation. In this step, you will have an assembly of proteins, a lot of proteins, tens of proteins, will assemble on one particular location, which is the +1 of a gene. And during the pre-initiation, the characteristic is that you don't have mRNA. Okay, the transcription has not started yet. But you have a PIC, which stands for pre-initiation complex. So these are a huge complex of multi proteins that assemble around the +1. But the transcription do not start yet. During initiation, and why it's do not start? Because you may have regulation. So sometimes you get assembly, but the transcription do not start. And actually what happens is that during the initiation, the DNA open. So you see here there is a kind of a bubble, okay? And you imagine here you need strength, because it's like a rope. So you have to put a lot of strength to open the DNA here. And then you have a small, short piece of RNA that is produced, which is under 10 base pair. And what happens is, most of the time, the transcription will fail. So this small piece of RNA detach, and start again, a new piece of RNA. And at some point, the transcription will start. So the elongation. During this more abortive, it's like abortion of small piece of RNA. This is regulated because you can have a factor that binds and will stabilize this small piece of RNA, and then you have a better transcription. During elongation, you have RNA synthesis. The critical part here is processivity, meaning that when the transcription has started, nothing stopped it. Okay, it's very like linked, it's locked. You have genes that are transcribed through the course of more than 24 hours. Like dystrophin gene is longer than a megabase. And even at 1,000 base per a minute, it takes more than 24 hours from the +1 to the end of the gene. Okay? So during 24 hours, you can imagine in the life of a cell, all the stimuli, glucose go down and everything. The transcription do not stop because the enzyme is really locked. But at some point, you have termination, meaning that termination is active. Since the enzyme is locked to process the transcription, you need something active to terminate transcription. Okay? And this is a signal. So you have a termination signal that actively end the transcription. So the RNA pol 2 will dissociate at this point, and the DNA will close again. And then you have maturation, meaning that the RNA, the mRNA that is transcribed is not the mRNA that you find in the cytoplasm. You have capping, meaning that the 5' end of the mRNA is modified because in the cells you have a lot of nuclease. You have endonuclease, you have exonuclease, and one of them is 5' exonuclease. So a piece of RNA, like this one, the small 9 base pair fragment, these are digested right away. As soon as they are produced, they get digested. Why? Because you have the 5' end, and there is an exonuclease that will recognize this end and will eat the small fragment. But mRNA, the maturation, do not wait until the transcription is over. It starts right away. Because as soon as you get 10 or 12 base pair here, you have a capping. A capping is an active process where the 5' end is modified by an addition of a nucleotide, but in the other way around. So it's not a 5' 3' link, it's a 5' 5' link. So it's a different link. Okay? And so what happens is that the exonuclease is not able to eat the mRNA by the end, because now this end is protected by the cap. That's why mRNA do not get digested just like that. Okay? They will be at some point digested because you want always a turnover. But it's active digestion, it takes a longer time. You have splicing, and again, splicing, you may have splicing of exon 1 with exon 2, even if the gene is not finished. Of course, it has to be long, because splicing takes a little bit of time. So if there is only, let's say, 2 exons, it will end and then splice. But if you have, let's say, 5, 6, 7 exons, probably exon 1 and 2 will be spliced long before exon 7 will be even transcribed. Okay? And then you have polyadenylation. What is polyadenylation? If you look most, not all, but most of mRNA, you have a poly A tail. So there is the 3', the last, if you look here, the last base pair. If you look mRNA, after that, you have a lot of A. Okay? So you have a special enzyme that add A at the end of mRNA. And this is to protect the mRNA from 3' exonuclease, because the same RNA will be eaten also by the 3'. Here there is not a cap at the end of a poly A. It's just the length of a poly A that allows the mRNA to be functional, because the A will start to be eaten, okay? But the coding sequence will remain the same. So you will always have the same protein. At some point, all the A will be eaten, then the 3' UTR, and at some point you are starting to eat the coding sequence, and so then the mRNA will be totally degraded. In this lecture, we will mainly focus on the pre-initiation step. It's the most critical, because if you don't have an mRNA, all the other steps are irrelevant, obviously. So the way you control the amount of mRNA is a starting point for all regulation. So as I mentioned here, you see, it can be done almost every single step. In biology, almost every step is always regulated. Okay, the mRNA, the amount which is produced, how it is degraded, then even the protein itself is regulated. All the steps are regulated. And here the transcription, so the pre-initiation, you have two opposite, and in a regulation in biology, it's always gas and brake. Okay, the best way to regulate something is to have two ways. You push and you pull at the same time. And it's the relative balance of both that will give you a perfect, well-controlled regulation. So here, as always, you have a gas. The gas is how do you choose the +1, and then regulation by activators regulation by co-activators. We will see what is that, but you see, this is the gas. And then you have the brake. The brake is, the DNA by itself, it's repressed. How can I say, the default state of DNA is repressed. So unless actively activated, it is repressed. There is no transcription. And why? Because the DNA is all packed, compressed, okay, in the structure which is called chromatin. And that makes sense because obviously with 3 billion base pairs, very few of that is mRNA. So it makes sense to have proteins that repress the DNA that do not require any energy, or except the energy to produce this protein. So when you produce here, you know, histone, you produce them, and then you pack all the DNA, then you do not spend any more energy. What you want to do is unzip tiny amount of this DNA and activate this particular. And depending on the cell type, it's not even all the gene, it's all a fraction of that. So on the energy perspective, it's very convenient to activate just a teeny amount of a genome. The actors. So now I gave you an overall shade. First of all, I wanted also to give you details, data about the actors that are involved in transcription. And here you have a couple of definitions that are very important. In biology, sometimes also you have names, and these names comes with a definition. And you have to stick with this definition. And sometimes you will see here some definition, how can I say, the name are not very clever, and I agree with that. But you have to deal with it. Okay? Like general transcription factors are not transcription factors. I didn't give that name, but I have to give it to you, and you have to learn that a general transcription factor is not a transcription factor. Every year I emphasize that, and every year then when I correct the copy, if I ask about a transcription factor, you can be sure that someone is going to talk about transcription TF2D, which is a general transcription factor. Okay? So the first actor, which is critical, it's obviously the RNA pol II, because this is the enzyme that catalyzes the transcription, okay, the reaction itself, which is polymerized ribonucleotide together. But if you, so like in the 60s, the first guy, not clone, because we didn't know how to clone, but purify the RNA pol II almost to purity, add DNA, that didn't work. And we know now that if you take DNA and you take RNA pol II, put them together, there is no transcription. It didn't work. RNA pol II, to work, require general transcription factors. So these are a bunch of proteins that are absolutely required to RNA pol II to function. If you put all together, that leads to basal transcription. So this is the minimum requirement to start transcription. Then you have transcription factors. Transcription factor, the definition is very easy. These are proteins that binds DNA to regulate transcription. They have to bind DNA. Then you have co-regulators. These are protein, also regulatory protein, that do not bind DNA. It's very simple. Okay? If you put all together, then you have regulated transcription. The way to see that is like a rheostat. Okay? When you have a lightning, you have a switch, okay, and then you have a gradation device. This is the switch. The transcription is on or off. Then this is the rheostat. You have a lot of transcription or you have few transcription. Okay? So these proteins by themselves are not able to start transcription. But these proteins by themselves, they are not able to achieve a normal, a physiological level of transcription. This is just to start. In other words, this, if you want to make it more simple, this is the PIC. So these proteins help to determine the +1 of a gene, assemble everything so the transcription can start. And if you let the system like that, it will start very slowly, make a few RNA, very few. Then if you add these guys, they will manage to have a nice, productive, very robust transcription. Okay? RNA pol 2. You see, that's not a protein, so it's several units, 12 units. And what is very important for in this, characteristic, it's the CTD, for C-terminal repeat. That is a 7 amino acid repetition, okay? Many, many times, up to 50 times. So the same 7 amino acid multimerized by 50 times. What is important in these 7, you have serine, threonine, and tyrosine. These are all amino acids that can be phosphorylated. If I remember correctly, it's 5 out of 7. So 5 by 50 times, meaning that into very teeny amount of the protein, you have 250 targets for phosphorylation. Okay? So it's very easy to understand that the RNA pol 2 will get phosphorylated or not. Okay? But you can see that on the GL, a huge difference in size. Okay? Then RNA pol 2 is as I told you, it's a polymerase. But you have different kinds of polymerase in nature. They are all defined by 3 different properties. Okay? You have, first of all, the choice of nucleotide. In nature, you have desoxyribonucleotide and you have ribonucleotide. So if you use desoxyribonucleotide, you are going to produce DNA. If you use ribonucleotide, you are going to produce RNA. So this one, it's a RNA polymerase. It produces a strand of RNA. Okay? Then you have to decide a polymerase. It makes a chain. Okay? Then you have to. The property is what kind of tempo does the polymerase use? The polymerase can use a DNA template. Like here, the RNA polymerase. It's an RNA polymerase DNA dependent because it makes a piece of RNA which is a copy of a DNA. Okay? But sometimes it can be RNA dependent. Okay? So you have some polymerase that use a piece of RNA to make a piece of RNA. So these are different kinds of viruses, as an example. Their material is not DNA. They keep as material RNA, which is copied as a strand of RNA. But some of them, you have also the other way around. So you have RNA polymerase, DNA polymerase, RNA dependent, like HIV. So you have an RNA within the virus, and that makes a strand of DNA. And then you have, let's say, a DNA polymerase, DNA dependent. So when you replicate your DNA, okay, when the cells divide, it takes the one piece of DNA, make another piece of DNA. This is a DNA polymerase, DNA dependent. And the last properties is the primer, because you have a nucleotide, 5 prime, 3 prime. The second one, it makes a link between the 3 prime OH with the 5 prime phosphate. They link together. It releases PPI, and you have a link. So number 2, number 3, everything works fine. The problem is for the first one. The first one has a 5 prime, but it requires a 3 prime OH to start. Okay? So this is the primer. Most of DNA or RNA polymerase require a primer. So it means it requires strand. This one do not require anything. So when you have a gene, I've shown you the +1. The +1 is added right away in front of the first one on the DNA. But let's say the DNA polymerase, when you make your replication, you need a 3' OH to replicate. This is a primase. When you do, I don't know if you have done already replication in the lecture, but you need a primase which makes the Okazaki fragment, which is a small piece of RNA. The small piece of RNA, the only purpose is to give a 3' OH so the DNA polymerase can function. Here's not very important, the biochemical path. This is how they have been isolated, verified. The important point is that you have several general transcription factors. It's not just one. You have A, B, D, E, F, and H. You see that you are missing C, you are missing G, G. These are because during purification step, they will turn out to be artifacts. So those ones are the real ones. After decades of work, we know that these are what you require, and only that. Most of these general transcription factors are not one protein. They are subunits themselves, complex. Okay? The most famous, I won't say important because they are all important. If you miss one of them, it won't work. But one of the most well-known is 2A2. Also, the nomenclature is very easy. Transcription factor 2, because they help the RNA pole 2, and then you have a letter. But you see, it's very confusing because their name is transcription factors, but they are not transcription factors. And first of all, why? Because they do not bind DNA. Most of them, actually all of them except one, all of them do not bind DNA. So they are not transcription factors. They are general transcription factors. And the name stands for the purpose, meaning that it was thought that these complexes are the same for the transcription of every single gene. Okay, it was thought that you add RNA code 2, this bunch of guys, and then transcription will start for any genes, which is about true. But the most important, as I told you, 2F to D. It's composed of one particular protein, TBP, which stands for Tata binding protein, and TAF for TBP associated factor. So TBP, among all of these, TBP is the only subunit that binds DNA. That's the only one. All other subunit complex do not bind. I think I have, yeah. This is how they are purified right now. Here at the RCM, there is a lab who is working with that. And I have another piece, just want to add something here. All of these complex, we will see that later, have a purpose by themselves. Okay, they are all have an important function. But what I really want to emphasize, but sometimes it's not so. Well, forget it. I will tell it later. I don't want to mix you up. General transcription factor, one piece. Then you have mediators and chromatin remodeling factors. Decades ago, these were not put in this category. Now that we know more about these proteins, we tend to put them here because they are not transcription factors as well, so they do not bind DNA, and they are a kind of general purpose. They do not have any kind of specificity regarding to a specific gene. They help transcription to a huge number of genes. Like the mediators is a subunit, a complex, big complex, that helps to be a link between transcription factors on one hand and the general transcription factors on the other hand. So meaning it's kind of link between an answer and promoter. Okay? And the way it has been shown, the existence, is through the squelching. Squelching is a phenomenon very important in transcription. It means that very often in transcription, when you put too much, it's not good for transcription.